Right, so I'm a hematologist by training, so just to give you a pre-warning, and I head up the Molecular Diagnostics Center, and so that's essentially, it's an interdisciplinary center of diagnostics. We're a bit of a hybrid lab, so we sit in the NHS, but we do a lot of translational research, and in that context I got very much involved with Genomics England. So I'm not employed by Genomics England or the Department of Health, I have to give that disclaimer, but I will tell you a little bit about the history behind the 100,000 Genome project, and I'm hoping you'll enjoy the talk. Please do interrupt me if I'm totally above your heads, or if you don't understand any slides, just warn me before I lose you. So okay, fine. So I'm going to head off, and I think what is important to understand first of all is that we've been thinking a lot about how we can improve patient outcomes, and a lot of the time when we think about how we improve patient outcomes, we're actually thinking about how we can improve treatments. And we're not so much thinking about how we can improve diagnosis. And when you start thinking about it, it's really important that you get the diagnosis right before you can then start to think about the right treatment. And it seems obvious what I'm saying, but actually very often in medicine it is not that obvious, and it depends also on how precise the diagnosis is, and a diagnosis might not mean the same for two different patients. So as you can see there, this is kind of 17th century diagnostics, and it hasn't really changed that much. To be frankly and open with you, we are defining disease by cause. So bit by a mad doggy is a cause, and we're still doing that in a certain sense. We are also, a lot of the diseases are descriptive. We basically say, okay, something is red or blue or whatever. So purple and spotted fever. There are still definitions that we use these days. How you die, whether it's suddenly or slow, again, it's something that occurs in medical classifications. And then when we really don't know, in particular in Britain, and I'm married to a Frenchman, and something is really horrible, we call it French. So it's the French pox, or so that gives you a flavour of how good we are in diagnostics. And this is 17th century, but has it really changed that much? So precision medicine is really about giving the right treatment to the right patient at the right time. And that's really quite important. And it's not just about the age of a patient, or it's not just about what type of treatment. It's also about the characteristics of the disease and the underlying reasons of that disease. So, and that really brings me to the molecular classification of disease. Now, what does that mean? So we can classify diseases according to lots of different things. We've just seen that. But in the 20th and 21st century, we've basically decided we have to look much closer at what is causing the disease, and then try and classify diseases according to their underlying molecular and biology. And so the reason why we are quite hopeful that we can do this now for a lot of diseases, not all diseases, but a lot of diseases, is because we understand the genetics of human genetics a lot better. And this is thanks to the fact that already now about 10 years ago, we in a big international effort, we sequenced the very first whole genome. And that means that we now know every single base pair of DNA of genetic material in your body, we know. And there are millions and millions and millions of them, and they all consist of 4 different letters, and I'll come on to that in a moment. So since this very first genome was published, it took years, and there's a picture of the machine there that was used. So that filled up more than this room. This was the first machine that did all the sequence, well, a large chunk of that sequencing. Thousands and thousands of patients have been sequenced of normal individuals, and what we primarily focused on is essentially sequencing their germline DNA. So DNA that everybody transmit to the offspring, and you do that by taking saliva or by taking a blood sample or a skin biopsy. The different types of tissues you can use, but essentially you'll end up with more or less a representation of what is in your ovaries or in your sperms, and then you're basically sequencing that. And so that's done on normal people. We've even sequenced the Neanderthal genome. So it's a very robust technology, and we know a lot more about now where we're coming from in terms of how humans and the Homo sapiens developed. We also know a lot more about, for example, about migration patterns and where Brits are coming from. So all of this information is hidden in the genome. Who killed somebody can be detected through genome sequencing. So there's lots of things we can do, but we can also do, and this is why as a hematologist I'm interested in, of course we can look at disease. And so the AML, the myeloid leukemia genome, was one of the first abnormal genomes that was sequenced. And that was then not using saliva or germline DNA, but it was actually looking at the genetic material in the leukemia. And I come on to that in more detail. So the machines have got really small. You can't really see how small they are, but they are benchtop machines now. And they don't cost lots and lots of millions of dollars, but you can actually sequence a whole genome, a human whole genome, for a little bit less than $1,000 these days. Obviously, there are other costs associated with it, but that gives you a rough idea of how feasible this is. And so in our laboratory, we've been assessing and evaluating different types of machines and different types of technologies to see how we can use them to really classify and reclassify disease. So before I go into this, I thought I'd give you a little crash course in biology and in genetics. This is a cell, and in the cell there's something that's pink, and it's a nucleus, and the genetic material is in that nucleus. So in the center of the cell, and it essentially consists of DNA. And if I can point the pointer. And so if you took a DNA strand and you really, you stretch it out, you will then identify different base pairs. And as I said before, they're symbolized by different letters, C, A, G, and T. And this structure that is this stretched out molecule actually wouldn't fit at all into this tiny little nucleus of each individual cell if it wasn't packed around lots of other proteins and really sort of compacted and compressed. And that is essentially what we then call a chromosome. So when you've heard of chromosomes, that means it's DNA, but it's kind of wrapped around proteins and packed really tightly so that it fits into the nucleus. And we've got 23 different chromosomes from a mother and 23 from the father, so that gives you 46. And that's how the maternal and the paternal chromosomes are coming from. That's where they're coming from. And the information that is in those DNA molecules will then be, in a very complicated way, translated by the cell into proteins. And proteins are essentially the bricks of the human body, and they can be enzymes, they can be structures, they can be useful for muscles, they can basically do all sorts of things. And they're really important. Now, what is important to remember also is that this stretched out structure here really is not just one single thing, but it has got lots of different functions. And some functions are there to directly encode the proteins, and some other functions are there to help with regulating the expression of those proteins going up and down. And so what we do in diagnostics conventionally, when we say to patients, you go to genetic diagnosis or you need a genetic diagnosis, what we used to do with the old-fashioned technology is we basically just took a very small piece here and we sequenced that. And so with the new technology, I don't know whether it's doing this, with the new technology, we can essentially sequence, the whole region of all these, chromosomes, and and we can do that really fast. And and this is what to some extent, what the 100,000 Genome Project is about. So I just want to give you a couple of examples now. So the first example is rare disease. Obviously, rare disease is not just one disease; it's lots of different diseases. And rare disease, the important thing to say about this is it's actually not so rare. This is a slide from the Department of Health. So when you look at this, 1 in 17 people will suffer from a rare disease at any point in their life. In the UK, we've got 3.5 million people suffering from rare disease. So that's an awful lot of people, actually. Only a quarter of rare diseases have actually had their molecular basis defined. So we know that they are very likely to have a genetic basis, these diseases, but actually we don't know what that genetic basis is. And that is not totally, but partially related to the fact that we so far haven't had the technology to sequence the whole genomes, and that we still have to do this on these patients. So there are 6,000 at least different rare diseases. Many of them are of genetic origin, already said that. And 75% of rare diseases affect children because of what I said previously. If you have a genetic disease and it's in your germline, then you can give it off to your offspring. So that is, can be, has quite a psychological impact on the family. And 30% of rare disease patients actually die before their 5th birthday. So that's, again, it's another important detail that highlights really the unmet clinical need. So, so then the question is, how do we look at rare diseases, and how can we, how do they present? And I'm sure you've heard about inherited diseases and that there are different modes of inheritance. I don't want to go too much into the details of it, but it is very important when you analyze rare disease patients with a rare disease, that you pay attention to the family history, and that you know whether mum or dad were affected, whether other brothers and sisters are affected, whether there are certain patterns of inheritance in their family. And very importantly for the rare disease program is that we're actually trying to focus on families where we've got several different programs agreeing to having their genome sequenced, because it helps tremendously in identifying what the underlying genetic defect is. So I don't want to go too much into the detail of that, but the rare disease program is really about getting families involved in identifying the abnormality. So there was an early example of this, and this is part of a precursor of the Genomics England program that was run here in Oxford, called WGS 500. So that stands for whole genome sequencing of 500 genomes of patients, primarily with rare disease, some cancers, that were recruited here in Oxford locally into this program. And so, and Professor Wilkie was able to identify in 2011, so already quite some time ago, a genetic mutation in a little girl, aged 4, and really that changed quite dramatically the family's outlook on this disease. Since then, we've had about 150 patients where we have the final data available, and this was published not that long ago in a nice journal in Nature Genetics. To cut long story short, overall, in patients who have gone through whole genome sequencing, especially if they've gone through this together with other family members, we were able to identify and make a diagnosis in about 30% of these families, where previously those patients had to go through all sorts of other investigations and have been sitting for years, thinking about their disease, and there was no answer. So I think we're at the beginning of making sense out of this, and 30% is not 100%. The main reason for why it's not 100% is because we still understand the biology of the genome not fully. And so it's a big focus of a lot of research labs in the university, is to when there are mutations in families, that we begin to understand what the functional impact of these abnormalities is. So an example from my world in hematology, we deal a lot with anemias, and obviously anemia is a very common problem, but then there are rare forms of anemia that are inherited and that very often present in babies. So we've had quite a few examples now where we were able to identify the anemia, and the important thing for the patients there is with this anemia is that actually it makes a difference to their own personal treatment. So in addition to being able to provide family counseling or prenatal diagnosis, we can also actually direct the treatment. And so in some patients it makes the difference between having tablet treatment or bone marrow transplant. So it has a very important impact on their subsequent management. So that's all I'm going to say about rare disease. The second example is cancer. And so you might say, well, why cancer? And so there are two different aspects to genetics in cancer. One is I call it the Angelina Jolie effect. So this is if you have got familial cancer, if you've got if you're a family and you've got cancer predisposition, then this is to some extent very similar to rare diseases, because it's in the germline, the abnormality, and you essentially sequence the germline and you do the family studies, and then you might be able to give out information to the family. But then there's a second aspect of that I'm as a hematologist much more kind of interested in, and that is how we can use the genetic information that is in the cancer cells or in the leukemia cells to direct treatment and to reclassify the cancers into different groups. So what is cancer? Cancer is, as I already said, it is essentially an abnormal growth of cells. And why do they grow? Well, they grow abnormally because they've got genetic changes, acquired changes in their DNA. And so those are changes that you don't transmit to your offspring, because they just occur in the cancer cells and not anywhere else in your body. And it's really important to understand that difference, because but cancer is an acquired genetic disease. So over the lifetime of a patient, the different tissues acquire mutations, and they are then specific for that tissue. And at some point, they acquire so many mutations that it will lead to cancer. So how do we classify cancer? And so this is going back now not to the 17th century, but to the 19th century. We actually classified according to the tissue of origin. So, when we say the cancer occurs in the brain, it's a brain tumor. When it's in the blood, it's a leukemia. When it's in the gut, it's colorectal cancer. And so the question is whether this is actually a bit too simple, because, of course, as I said at the beginning, where this comes from is important, but it's not the whole story. We have to understand, what is actually the cause and where the genetic changes. So this is what pathologists are doing these days, and I do as a hematologist. So we take pretty pictures and we stain, the tissue that is coming from those tumors. We stain them up with pink colors, and it looks very pretty. But does it actually tell us whether a patient will respond or not respond to treatment? And most of the time, it doesn't. There are exceptions to the rule. And yes, there might be some patients with lung cancer that respond better to certain types of treatments than patients with prostate cancer. And we certainly have differences in the different types of leukemia in terms of responsiveness to treatment, just looking at the morphology. But really, they are not very good discriminators. This tissue classification is not a very good discriminator, and is certainly not the only pillar of precision medicine, or can't be. So the other problem we have with cancer is that it's, genetically speaking, it's a moving target. So what happens with cancers is a bit like what happens with bugs when they become resistant to antibiotics. And that's a concept I think that people are familiar with. With cancer, it's maybe not that known. But when you get these gene changes happening inside your body and you then develop cancer, then that cancer won't stay the same. It actually changes and it mutates. And when you give a treatment, initially the patient might respond, but then the patient becomes resistant to that treatment. And that's because there are new mutations occurring all the time. And it's exactly the same as what Darwin described for the first time in Galapagos, with the beaks of with the beak structure of the finches. So that cancer adapts to its environment, and if the environment is a certain type of chemotherapy, well, that's great. Cancer adapts to it because there are these mutations occurring all the time, and they will make the cancer survive. So that's basically, temporally speaking, it's a moving target. It's also spatially speaking, it's a moving target. So if you have a big colon cancer, for example, and you have metastases, you've got spread of the cancer in the lung, and you take different tissues from the lung and then from the cancer in the gut, you will find that those are different. Some mutations are the same, but a lot of them are different. And so there's a lot of what we call heterogeneity in cancer and in the genetics of the cancer. So it's a moving target, and that's what's making it particularly challenging to treat. Even within one biopsy, we find lots of branches going off of lots of different cells that are genetically slightly different. in hematology, I claim that we have for the first time actually applied precision medicine, and that was a German, surprise, surprise, called Vertov, and he essentially described for the first time very precisely one type of leukemia called chronic myeloid leukemia at the middle of the 19th century. And following on from there, and it's a very interesting story, if you do have time and you're interested, you can read it up. There's lots of information on chronic myeloid leukemia, because it is a simple disease. It's a simple leukemia. There's one genetic change, and it's that genetic change that drives the leukemia, and it doesn't really change that much over time. So thanks to that, it was the first the leukemia cancer where actually a genetic abnormality was described at chromosome level. So at this sort of compact DNA level, and it was called the Philadelphia chromosome because it was described in Philadelphia in the 1960s. It was also the first malignancy and cancer where then actually the underlying molecular structure of this abnormal chromosome was described, and it's called the 1922 translocation. I don't want to go into this, but Janet Rowley spent a lot of time in Oxford, so doing cytogenetics here. So there are links there, and then it was one of the first leukemias that were actually treated successfully with either radiotherapy or then subsequently chemotherapy, busulfan hydroxyurea. Then came bone marrow transplantations. For the first time, patients with cancer and leukemia were cured with bone marrow transplantation, and then it was the very first leukemia and cancer where actually targeted treatment, so treatment that is not chemotherapy but directly targeted towards that genetic abnormality, was very successful. And so these days, patients with chronic myeloid leukemia are no longer having a bone marrow transplant. They actually take a tablet a day and they are fine. And they're fine for years and years and years. And it's totally changed outlook of this disease. So this is partly because it's an easy disease, because there's only one genetic abnormality, you target it successfully, and patients do no longer die, and they're fine for as long as they take the tablet. So in cancer, proper cancer, not blood cancer, but in proper cancer, this is an example of skin cancer. As you can see, it's a lot more complicated because melanoma, they don't have one genetic abnormality. We know from the whole genome sequencing projects they have got 20,000 on average. And so and then they change over time, and they change spatially. So when you look at all the different nodes this patient has got it under the skin, the infiltrates, they're probably genetically all different. Not completely different, but a bit different. Different enough to cause resistance. So what we do with targeted treatments now, a few years ago, we basically then discovered that there were some mutations that are particularly driving disease progression in patients with malignant melanoma. And in this case, it's something called BRAF. You don't have to remember the name, but it's just a gene. It's just a mutation in a gene. And some clever pharma industry then came up with an inhibitor of BRAF. And so they did these studies, and it took them 9 years to develop the drug. So really targeted, looking at the mutation, how that changes the protein, how you then develop a molecule, a drug treatment that prevents this BRAF from being active. And you can see the effect, in panel B. You see the patients responded really, really well. So everybody was thinking, oh yeah, we cracked it. And then obviously 9 months later, patient comes back, they all come back with the same problem. And so they relapse because the tumor becomes resistant. They also develop other side effects, they develop other cancers, and we don't really understand well why that is. But it tells you it takes 9 years to develop a drug against a single genetic abnormality, cost millions of pounds, and 9 months after you're back to square one. So on the leukemia side, when you've got single abnormalities, that's really great and it works. On the other hand, with solid tumors, when you've got thousands and thousands of genetic abnormalities, actually it doesn't work that well. But it works a bit, and so that's a start, and we have to start somewhere. So there are many ways to kill cancer cells. So targeted treatment is one of them. Obviously the ones that we're all familiar with are surgery. Put the knife there because I'm not a surgeon. Radiotherapy. And those two modalities, although I'm not a surgeon and I'm not a radiotherapist, oops, they cure most of the patients. So if you have got a cancer that can be removed surgically, that's really good. If it can be completely removed, that increases your chance of cure. If you have got radiotherapy, again, you know, that is a really, if you are offered radiotherapy, it works, for most cancers, it works actually quite well. The chemotherapy, and that's where I actually work in the chemotherapy field, actually doesn't work that well. It works okay-ish for leukemias, but for solid tumors, often it doesn't work that well. So how does this work? You know, so chemotherapy essentially, and this is kind of a cell. So this is a cancer cell. Inside there's the nucleus, and inside the nucleus you've got your genetic material. So the chemotherapy just, you know, induces a lot more mutations into the genetic material, so that the tumor really can't cope anymore and it just dies. That's the theory. There are some people saying now, and there's some evidence for that in research literature, that if you give chemotherapy, sometimes that can actually induce more mutations that then make the cancer even survive better. And so maybe giving chemo is not always the best thing. So then the other things that you can give is the small molecules inhibitor, and these are things like BRAF inhibitors, because they interact directly with the signaling and from the outside to the inside. And every cell, even a cancer cell, needs signals coming in, survival signals coming in from the outside, going into the inside. And that's those signals are coming from the microenvironment. So wherever the cell lives, that's where they are. So if you can switch those survival signals off by giving small molecule inhibitors, either in here or by using antibodies that switch off the contact here with the microenvironment, you can potentially kill off the cancer cell. And then another very interesting kind of treatment approach that's been around for a long time in hematology, and bone marrow transplantation is one of them, is immunotherapy. So the idea there is essentially that you're using the patient's own immune system, or in allogeneic bone marrow transplantation, you're using somebody else's immune system to kill off the cancer cells. And if you've got very few cancer cells left, because most of them have been killed off by chemotherapy and there are only a few left, then this can be very effective, because the immune system can like it detects infections, and it can detect cancer cells and it can kill them. And so that's something that is very interesting, and I'm sure you've all heard about how stress can cause cancer. So there are these funny links between the immune system and cancer. And in the recent years, and this was in the big press several times now, is that what people have realized is that a lot of the time when patients get cancer, these cancer cells, they express certain proteins on their surface that basically mask them against the immune system, and the immune system can no longer recognize the cancer cell. So what the clever treatments can do, clever antibodies can do, they can basically bind to these are the antibodies here, they can bind to these proteins on the cancer cell surface, and they can unmask the cancer cell, and so that it's recognized by the T cells and the dendritic cells that are really the immune cells in everybody's body, and then that leads then to death of the cancer cell. So these are very, very promising agents, and I'm sure that you will hear a lot more about them. And generally speaking, people are talking about immune checkpoint antibodies. So we've got all these different things and all these different ways of killing off cancer cells. And so the question is then, what patient should get what treatment? And there are obviously ways of tailoring treatments, lots of different ways, clinical ways, saying younger patients should get this, older patients should get that. Some patients tolerate better this treatment, older patients, or some other patients tolerate better that treatment. But then it comes back also again down to the genetics of the cancer. So what we did in our molecular diagnostic lab is that we've basically designed within for NHS patients a rapid test that is genetically based and that can identify lots of different mutations so that these patients can either get access to drugs through the Cancer Drug Fund, or they can access it through a clinical trial if there is one. So this has been up and running now for the last 2 years, and it's been quite successful. And it doesn't have the same cost as the whole genome, where we're still talking about roughly $1,000. This is a test that is £300 and that has a 6-day turnaround time, so it's very fast. So coming on to the 100,000 Genome Project now. So I've given you a little bit of background of why David Cameron came up with this brilliant idea of investing £180 million of public funding into sequencing 90,000 patients with rare disease and cancer. So that's the background behind it. It's a brave move because there's still a lot of uncertainty around how useful this actually will be. On the other hand, there's certainly tremendous interest within the research community because it will will increase our understanding of these diseases. So the aims of the 100,000 Genome Project is then to identify the best of existing treatments for patients, and we've discussed that, how we can design or direct treatments according to mutations, to provide prognostic information. And prognostic information doesn't mean that we know what is the best treatment, but at least we can say to patients, okay, you'll be doing fine, whereas you might not be doing that fine. And for patients, this is important. It's a difficult conversation to have, but if, for the clinician, but it's actually I think really important, and it's what we hear from patients is that's often what they want to know, and sometimes they don't. It's also important that it provides a big data set to allow researchers to better understand the biology of the different cancers. So all the information will go into a big database, and that in an anonymous way so that researchers can access it. And then it will provide UK academia and pharmaceutical companies with information about future therapeutic targets. So essentially, by having all that genetic information, we might find new treatments. That's essentially what this is about. So how does it work in practice? And this is a English project at the moment, so it's called Genomics England. There are other initiatives going on in Scotland, very similar. There's a Welsh idea of having Genomics Wales. Genomics America has been announced by Barack Obama. The Chinese are busy sequencing away. So there's lots of sequencing similar activities now going on in other countries. So we're not the first, but we are one of the first. And it's essentially the advantage I think England has is that we have got a national health service, and that because you need large numbers of patients in order to make any sense out of this. And so in private healthcare system it's a lot more complicated. So what this means practically is that if you are a patient with rare disease or maybe cancer patient, you might be approached at some point, and there are criteria around this, so it's not you who chooses or your clinician who chooses. There are strict inclusion and exclusion criteria. You might be approached to undergo whole genome sequencing, and then you go through a consent process. The samples will be prepared, and then the data, both your clinical data and also the genome data, will be stored inside the NHS firewall. So it's not something that will go off to a third party or to God knows where, but it will be kept as safe as possible. The data center for the Genomics England project is the same place as the Medical Defence department, so the Ministry of Defence in Wiltshire. They are, it's a very safe place. Obviously, anybody can hack into, not anybody, but some people can hack into the US Ministry of Defence, so I'm not saying it's safe, but it's sort of as safe as can be, probably. The important thing with this is that it is not a normal kind of research project, but is actually a project that should transform the NHS by giving the NHS the capability to do whole genome sequencing, and then importantly to at some point feed the information back to the diagnostic services in the NHS so that the patients can benefit from the information. And the way the information will be fed back is via the clinicians. So it's not the you don't get a letter through the post saying you've got 20,000 mutations. It goes back to the clinicians, and then the clinician will hopefully be able to explain the results to you. So that's what this looks like. It's a very complicated process because it involves, it's England-wide. So which cancers will be primarily sequenced at the moment? These are the ones that have been identified. Sarcoma has just been announced, so has kidney, and then there's the cancers of unknown primary. So the list of cancers at the moment is still enlarging. It's not going to be eternally enlarged because statistically speaking, we have to have at least about 2,000 genomes per cancer type before we can actually make any kind of clear get any clear results. And then what we're going to do is we're going to take the germline sample with all your germline variants, from the saliva primarily, or from a peripheral blood sample, depending on which tumor it is, and then we will look for the G there, for one single mutation in all these different base pairs. We'll bioinformatically subtract, one sequence from the other to make it a bit easier. So this is not something that can be done overnight, and it's something that requires bioinformaticians, clinical scientists, people who are really specialists in diagnostics, clinicians, oncologists, the clinical geneticists on the rare disease side, genetic counselors. It's a multidisciplinary project, and it really requires everybody to work together, including also the researchers who, to a large extent, will make sense out of the data. So what are patients thinking? And this is Jane Niederer, who's the research nurse who is leading on this project from a research nurse perspective at the moment. So she's involved a lot with consenting patients. So what information about my cancer treatment will be fed back to me? As far as cancer is concerned, primarily really only the information that is important for treating the cancer. There's a very short list of other actionable mutations that would occur in the germline and that would be of relevance for your family, but mostly it's just information that would be important to treat your cancer and to solve that problem. Will my life insurance be affected? No. So there's a law at the moment that basically says that that your life insurances can't access any genetic information. It's up for review, this law, at some point, but I think everybody expects that it will be the same and that insurance companies will not get access to this data. If DNA is specific to me, how will my identity be protected and anonymized? So the names will be taken off, your postcode will be taken off before anybody can access the data, but clearly your genome is unique, unless you're an identical twin. So theoretically, there is a way of knowing exactly, what your genome looks like, and so you can identify, patients on the basis of their genome. Question is how interesting that is, and whether people really want to hack into, a major a data center to dig out, Mr. Smith's genome. So that's a question that that I think every patient has to answer for themselves, and it's all to do with how we manage big data and how we manage the internet and how we manage all these, these problems. How long will I have to wait to find out relevant information about my cancer treatment? So that's an interesting question. We haven't answered that yet for the Genomics England project. Obviously, you can't wait too long because cancer is a life-threatening disease. So that needs to be that question needs to be answered. Will the findings affect my children? Well, we've discussed that. Sometimes they might. For cancer patients, mostly they won't. If it is a rare disease family, yes, there's likely that it will, but we will offer genetic counseling, and you will be able to discuss, the impact of the findings, with the genetic counselors. How do I withdraw from this study? Well, anybody, some of you might have been part of studies in the past, anybody who's part of a study can withdraw at any time. What you have to do is you ring the number and you say, I don't want to be part of this anymore, and then we destroy all the information and we delete you from the database. It's actually very simple. What is the future? And I just want to give you a brief outlook of this. So you saw the machines at the beginning, when the first whole genome was sequenced, the machine was so big it wouldn't fill the room. It would fill the room. Then now we've got benchtop sequencers. They're pretty expensive. They cost around £500,000 each, these machines. That's not going to be like this forever. There are now several companies and they're heavily competing with each other, including some Oxford spin-outs like Nanopore. They will provide, in the not too distant future, similar but different technology so that you can probably sequence a whole genome, on a device that is not bigger than a mobile phone. And they will probably do this, at a cost of $50, and it will be very cheap, very, very fast and very cheap. At the moment, this is not, it's not that precise yet. But what is already happening is that there are clinical trials ongoing to look at diagnosis of tuberculosis bugs in developing countries, in emerging countries, where bugs have got a smaller genome, so it's easier to sequence. So those sort of applications are already ongoing, and it won't be long before the first whole genome will be sequenced on a small device like this. Sequencing of plasma DNA is another interesting topic. So this is just a patient with malignant melanoma, again, who had metastases. I won't go into the details, but he had lymph node metastases in the mediastinum, and it was impossible to biopsy these nodes, and that might be important, of course, when you're thinking more targeted treatment. So what people can do instead is taking a blood sample and looking for circulating DNA of the tumor in the blood sample. And that's probably the way how we're going to take tumor sequencing forward, so that patients don't need biopsies all the time, but they can, and invasive procedures where you have to stick needles into people or even undergo surgery, but you can actually detect the circulating tumor DNA in the plasma of the patients, and then you can diagnose the cancer that way. You might be able to do early diagnosis in that way, and you also might be able, like in this case, to track response to treatment. So you can see from this here that there's a melanoma patient, lots and lots of mutations initially. He then received treatment and he responded really well to the treatment. And you could see that his nodes got smaller, the melanoma on the skin was controlled, everything was fine. And then the clinical relapse was essentially diagnosed at this point here, whereas we using the tumor DNA, we were able to detect that he was relapsing and becoming resistant to the treatment several months earlier. So it's a way of monitoring response to treatment, and maybe at some point in the future, not at the moment, but in the future maybe changing treatment on the basis of the information we get from the circulating tumor DNA in the plasma. And that is done on a blood test rather than an invasive procedure. So the immune system, we've already discussed it. The question is, who's going to respond, who isn't going to respond, and the interesting kind of thing that is emerging now is that the more mutation a cancer has, the more likely it is that the immune system will recognize it as foreign. That kind of makes sense intuitively. And so again, if you're able to count all the mutations across the whole genome, like you can do with whole genome sequencing, you might be able to identify patients who would respond really well to immunotherapy. And so that's something, again, that is a very active area of research for a lot of people at the moment. So, in summary then, where this is going is basically that we are going from more symptom-based more symptom-based kind of intuition medicine or evidence-based medicine even, to a more precise medicine and maybe algorithm-based medicine. Now, I'm a clinician. I don't really believe 100% in algorithms, but there's an awful lot of information that we have to take in about not just the genome, but other characteristics of the disease. And if we want to make sense out of it, we really need the help of computers. So it becomes a little bit similar to all the other big data initiatives that are ongoing, including Facebook and Google, where you have to really take lots of information from lots of different places and then make sense out of it. And so in the university we have got the MDC, so the Molecular Diagnostics Center, where we basically generate lots and lots of different data. We then have the Oxford Big Data Institute that is currently in construction. You may have seen on the Old Road campus where the big cranes are. That's where the Big Data Institute will come up. And in there will be lots of clever mathematicians and statisticians who will make link up all these different data together with the clinical data. And then in the Oxford Precision Cancer Medicine Institute, that will hopefully go up onto one of the car parks and make, then we have even fewer car parks on the Old Road campus. But essentially, that Precision Cancer Medicine Institute will focus specifically on doing precision medicine using genomic testing and other testing to then target the right treatment to the right patient at the right time. So in summary then, I think it's an exciting opportunity to perform sequencing on large numbers of rare disease and solid tumor patients. We will have the opportunity to collect longitudinal data, in particular also clinical data, and to improve the quality of the clinical data that is currently in the NHS for research purposes. We'll be able to feed back results that are validated, obviously, through to the clinicians, and then hopefully patients taking part in this will have opportunity to benefit. I didn't really talk about this, but as part of the Genomics England program, we're not just collecting samples from the tumor, but we're also collecting, and not just DNA, but we're also collecting other samples, including those plasma samples. And then we also look at protein expression and RNA expression. So there's other samples that we collect that will be useful for researchers primarily. The data will be available to researchers and to industry, and also to the, obviously to the NHS. And so I think it'll be an opportunity for, on a wider scale, to really get the NHS working together with the universities and the researchers. So it's a very special program in that respect, because it doesn't always happen that we're working in an integrated way between academic research and the hospitals. So I'm going to stop here.